Hello everyone and welcome to our YouTube channel Meet You. In today's video lecture, we will be doing an important concept from the molecular basis of inheritance, the transforming principle. Before understanding this principle, let me tell you something about one type of reproduction which is seen in bacteria and is called as the transformation. Now, transformation is a type of a type of transfer of genetic material, not really exchange but transfer of genetic material. Here we have a bacteria. This particular bacteria will pick up naked DNA. This is just the DNA and it is referred to as the naked DNA because this DNA was the result of the lysis of another bacteria. So this particular live bacteria will pick up this naked DNA and will incorporate this naked DNA into itself. And this incorporated changes may at times bring about certain transformation in this bacteria, resulting in the formation of certain features and certain structures which are associated with the naked DNA. This is called as transformation in bacteria. The transforming principle which was given by Griffith, the most important principle lies in this process of transformation. However, you have to make a note here that at times during the process of transformation, at times there are certain features where the bacteria may not accept the naked DNA which was transferred from the external medium to the bacteria. In that case, this bacteria may produce certain kind of enzymes, which nuclease enzymes, which may result in the lysis of this naked DNA, thereby maintaining the integrity of this bacteria. Let us see how Griffith tried to explain this transforming principle. The transforming principle also deals with, Griffith was trying to, he was trying to explain that it was the DNA which was actually responsible for the transformation of all the characters in the different strains of bacteria. Griffith was doing experiment. He was carrying out experiment on pneumonia, and in that process he cultured two different strains. We have to remember two different strains of bacteria were cultured. Two different strains of bacteria were cultured by Griffith. The two different strains had various characteristic features. So he observed, let us see. We'll name this as strain 1 and we will name this as the strain 2. He observed, he had a very good observation and he observed that the strain 1 had a very smooth, shiny, very smooth and shiny surface. So it had a very smooth and shiny surface. So he named it as the S strain. The second strain did not have as much shining and smooth. The surface was quite rough without any smooth or shiny feature on the surface. Since the surface was rough, Griffith found it appropriate to name it accordingly as the R strain. Now, he found two different strains, S strain and R strain, based on the presence of the surface that was produced in the culture medium, in the petri dish where the culture was carried out. However, Griffith, even after that, was not able to determine which strain was actually resulted in causing pneumonia. He did further experiment. The most famous experiment that we learn in the chapter Molecular Basis of Inheritance. We see here in this chapter that when Griffith took mice and he injected different strains of bacteria and he carried out his experiments. Let us see further what were the different steps of the experiment which was conducted by Griffith. We rightly call this experiment as Griffith's transforming principle. Let us see what this transforming principle was. Now we have to understand this. Griffith wanted to make sure which strain of bacteria caused the disease or caused pneumonia. He had two strains. He was doing experiment on the two strains, S strain and R strain. So to determine that, he conducted these two steps. In the first step, what he did was he took up the S strain bacteria, injected into the mice, and what he observed was the mice died. On conducting autopsy, he found that the mice suffered from pneumonia, and of course he injected the S strain, so we found traces of S strain. But still he did not conclude that S strain was the one which is pathogenic or caused the disease, because he still had another strain, that is the R strain. So next what he did was he took the R strain and he injected the same to the mice, and he observed that the mice lived. This particular step, these two steps confirmed that the S strain which was cultured in the medium he had, in the petri dish that he had, this is the pathogenic strain of bacteria. Pathogenic strain, as we all know, the pathogenic bacteria that causes the disease. Okay, that causes disease. Those bacteria called as pathogenic bacteria. We also have one conclusion. We know that pathogenic bacteria are capsulated bacteria. They are capsulated bacteria. So he easily concluded that in the petri dish, the smooth surface was the result of the capsules which were formed by the bacteria. So it rightly concluded that the S strain is a pathogenic form, whereas the R strain had a rough surface. Okay, it had a rough surface, indicating that it is non-capsulated. Also, when the mice was injected with the R strain, the mice lived, indicating that it is non-pathogenic form of bacteria. So these two steps concluded which is pathogenic and non-pathogenic. Let us see try to understand why the disease was caused. Now, any capsulated bacteria, definitely if any bacteria is able to cause disease in human, in animals, in anything, if it's able to cause disease, means that the DNA of the bacteria has got certain information which will bring about changes in the genetic machinery of the host and cause the diseases. It has information of the disease, and that's why the disease is caused in the host. So definitely the S strain bacteria, okay, so the DNA which was present in the S strain bacteria, the DNA, the genetic material, okay, genetic material in the bacteria, the S strain bacteria had the information for the disease, okay, for the disease. Therefore, the mice died, okay, due to pneumonia. The reason behind why Griffith conducted these two steps in the initial stage was Griffith wanted to confirm which is the pathogenic strain, that means which causes the pneumonia, which causes the disease in the mice. So these two steps confirmed which was the pathogenic form or the pathogenic strain of bacteria. But however, there was another point Griffith also wanted to he wanted to convey that He wanted to say that DNA is the genetic material and DNA itself has all the information. So to carry out further his experiment, to understand and try to give conclusion to his experiments, he did further few steps, and let us see what are the few other steps that were conducted in the transforming principle by Griffith. Griffith stated that the S strain is a pathogenic form. Let us now see the remaining steps of the transforming principle, and let us see how Griffith tried to explain that DNA is the genetic material. So what he did was, he took the S strain bacteria. Now he heated the bacteria on, this is the S strain. Let us see the genetic material, nucleoid of the S strain. So what he did, he induced heat. On heating the bacteria, all the structures of the bacteria lysed and the bacteria was dead. However, the DNA or the genetic material remained. The DNA of the genetic material remained in the medium. You have to remember this. It remained in the medium where the heating was carried out. Remained in the medium where the heating was carried out. This point is very important. So now when we are talking about the DNA material which remained, so this was, what did Griffith do? He took a sample of the medium separately. He took a sample of the medium separately, and then he carried out his next important step. In the next step, what he did was, he took the heat-killed bacteria, or let us be more specific, the heat-killed S strain. Heat-killed S strain. Okay, so you have to remember, it is the heat-killed S strain, and he injected this into the mice. The observation that he saw was the mice lived. Okay, mice lived. Alright, mice lived. Now, this conclusion was a big surprise to him. Let us see the concept behind this particular step. We know that the S strain is the capsulated form. It is a capsulated form. So, heating removed the capsule. Heating removed the capsule, and as a result, it lysed the bacteria. It lysed the bacteria. So only what was remaining? Only the DNA was remaining. So since only the DNA was remaining, this DNA was not able to kill the mice. It was not able to kill the mice. Neither it was able to cause the disease, because the DNA did not receive a compatible host where it can carry out its function and cause the disease for which it has the information. That is why the mice lived. Now Griffith conducted the fourth step, okay, which was an important part or an important step of this transforming principle. In the fourth step, Griffith took, let us be specific here, he took heat-killed S strain bacteria, heat-killed, you have to remember this, heat-killed, and he mixed it with the, let us see here, as I told you, specific live R strain bacteria. This indicates that one is the killed one, another is the live one. Alright, and then he took this mixture medium and injected into the mice. The observation he got was mice was dead. Mice died because the mice suffered from the disease, that is pneumonia. Then he wanted to make sure how did the mice died. Okay, how did the mice, because he knew that in the previous step he injected this heat-killed S strain into the mice, but mice lived. And then we have the live R strain, which is of course non-pathogenic. Then how come the mice died? So he took up the autopsy sample, and to his very surprise, he found in the dead mice, he found traces of live S strain bacteria. This was an important step, an important conclusion in Griffith's transforming principle. Now see, he injected killed S strain and injected live R strain. But what we got? Live S strain bacteria. Let us see the concept behind what happened in this step. Now, as I told you, transformation. This is the R strain. This is the R strain bacteria. And this is the DNA. Let us see, this is the DNA of heat-killed S strain in the medium. Okay, in the medium. So what happened? Now, the DNA of the heat-killed S strain and the R strain were brought together in the same medium. They were brought together in the same medium. You have to understand, they were mixed together in the same medium. Due to the mixture, this R strain was able to pick up, or the DNA of the heat-killed S strain was able to get absorbed in this live R strain. When the DNA was in the R strain, okay, now this R strain this S strain DNA got incorporated into the R strain bacteria, thereby controlling the machinery of the host. Now you have to remember what is the information that is present in the DNA of the heat-killed S strain. The information that is present, point number 1, it has the information to form capsule. Point number 2, it has the information of causing disease, that means pathogenic form information. So these two informations were incorporated in the R strain, and the R strain was able to form a capsule. First information, okay, due to the information of the DNA here, it is able to form a capsule. So this is the capsule, it's able to form the capsule. Secondly, this DNA transformed this non-pathogenic DNA into pathogenic, thereby converting this entire R strain into the pathogenic form. Okay, converting it into the pathogenic form. So inside this bacteria which is there, it is R strain only. It is not S strain, it is R strain. But since it has the DNA of the heat-killed S strain, and the S strain DNA has got information of these conversions. So what it did, it formed a capsule. Okay, suppose this is the R strain, it formed a capsule around this. So it became capsulated. Okay, it became capsulated. Secondly, the information of the disease causing information was there. So this R strain was able to cause the disease and thereby kill the mice. But however, it was the R strain bacteria only. But since it was capsulated and it formed and it was able to cause the disease, Griffith found live S strain bacteria. Even after this step, Griffith, however, was not convinced and was not able to conclude that DNA only is the genetic material, and it was the DNA which was actually carrying out all the changes. So his experiment formed a base, formed an important part of the study, that DNA is the genetic material. Okay, so this was all about the Griffith experiment. 16 years after Griffith's transforming experiment, the actual conclusion came into view, which was given by the biochemical characterization of the transforming principle by MacLeod, McCarty and Avery. They used different kind of enzymes which were used to lyse the different parts of the bacterial cell. They use proteases, they use lipases, they use RNAs, and finally when they use DNAs, they were able to conclude that it was the genetic material that was able to actually cause all this transformation from the R strain to the S strain. That means it was the DNA which was actually responsible for transforming the formation of capsule, information for the formation of capsule and information of pathogenic form to the R strain and then converted into the S strain. Another conclusion was given by Griffith. After performing the entire steps of the experiment, what Griffith did was he cultured the transformed strains of bacteria, and on culturing the transformed strains of bacteria, he found that even the progeny possessed the transformed information and were pathogenic in nature, indicating that the transformation that took place in the R strain was actually a permanent change.